Dynamics. Oh, darn. Of nuclear, including the structural dynamics of the nuclear pore complex chromatin and mechanotransduction between the nucleus and the cytoplasm. So please join me in welcoming Dr. Elizabeth Villa as soon as we figure out our audio situation.
All right, fingers crossed, we should be good now.
All right,
Can you hear me?
amazing. We can hear you. Can you hear us?
I can hear you. That's great. And I can see you. I see you have an owl, which I love, or
We
a similar
do, and
camera.
if I hit the right button, we will record.
I think someone asked me to record, and I think it's already recording.
All right, awesome. Thank you. Everybody give a round to Marcus. Thank you.
All right, good.
I'm so sorry, Dr. V. I apologize.
Oh, no, I'm the one who's sorry. I'm sorry. I wanted to
Try
come
real hard
and
not to be late. So
show you the dog. I'm dog sitting this week, but unfortunately my daughter also had plans to hang out with the dog and she's sick. So she's hanging out with the dog. And I'm here. So yeah, I wanted to tell you a little bit about the work in my lab. As you heard, there's a lot of different areas in my lab, and I'm just gonna tell you a little bit about the methods that we use, and then one story that's a lot of fun in the lab. They're all a lot of fun, but this one's particularly fun. It also spans a lot of different labs. I use San Diego, so it's like super cool because we're very collaborative and really kind of like spanning from atomic models of proteins and molecular dynamic simulations of those models all the way to sort of like organisms, and as you'll see, even clinical studies at the end. So talk about interfaces. It's a really fun thing. All right, so I sort of like I have a dream slide is a little bit about, I think most of you probably have heard that cryo-EM took structural biology by storm. And I think it's safe to say that most labs, or almost every lab who's doing structural biology in any shape or form, either is already doing cryo-EM or trying to learn how to do cryo-EM. We underwent something called we call it a resolution revolution about 10 years ago with the development of new cameras. And, it really is now relatively routine that if you can biochemically isolate a protein, it is likely that you're going to be able to get its structure or a macromolecular complex. So now we know that electrons are the best radiation that we have right now to look at biomolecules, but this doesn't have to be only isolated sort of like outside the cell. It can also be inside the cell. And I've sort of dedicated a little bit more over a decade to making this happen. And now that it does, to use it to a diverse, set of fun biological problems. And so the thing about typically about imaging biomolecules is that in life, just like in imaging biomolecules, there's no free lunch. And so typically when you want to look at very, very high resolution structures, so that you want to see a catalytic side, or really like the atoms of how a drug binds into a protein or things like that, typically you're looking at what we call structural biology techniques like single particle cryo-EM, X-ray crystallography, NMR, and those techniques, where you basically do that at the cost of removing the proteins from their natural environment, which is the cell. So a little known secret is that any given time, a protein is actually interacting with a complex of 10 other proteins on average. And it does that, like a lot of proteins actually interact with many of these complexes or become part of many different complexes in their life cycle. Just the same way as that, you see your cat in the morning and then the barista at the coffee house and then your lab mates or whatever, right? So similar people in class. And so when you take away this context, you can learn a lot about these proteins, but it's not the whole thing. And sort of like this molecular sociology that I'm talking about, it's really more the realm of cell biology and the other side. And for cell biology, we typically use, to see how these proteins interact, where they do it, when they do it, and so forth. We use light microscopy techniques that sort of have spun into this very powerful super resolution light microscopy, but also electron microscopy really started the field of molecular cell biology, sort of like 60 years ago or so. But somewhere in the middle is cryo-electron tomography that I'll talk to you about today. And we and others are trying to push the boundaries in these methods. It's both a bona fide structural biology technique, but also really looking at the structures of molecules within the context of their natural environment of the cell. And so if you have a cryo-EM, like we have many on campus, both in engineering and in biological sciences, depending on your sample, you can use the microscope in different modalities. The more famous one that you'll probably see constantly in the cover of nice journals is single particle analysis, where you purify molecules and you basically make a monolayer of these molecules embedded in ice. And if you're lucky, they're captured in a lot of different orientations. And one image from a transmission electron microscope is literally like the electrons that get transmitted. And to a first approximation, it's proportional to the mass of the object. So like high intensity pixels are like high, or other low intensity, whatever. It's high mass areas, right? So heavy atoms give you darker pixels and so forth. And it's sort of a projection through the entire sample, exactly the same as an X-ray. So it's not just sort of, so you basically have a projection through the entire sample. So if you catch enough images in enough different orientations, basically you can reconstruct them in 3D. And that's sort of single particle analysis. Then you have also this electron crystallography, where if you can make crystals, you can actually use the microscope in diffraction mode. You shoot it at it, it sort of diffracts electrons in particular orientations, and you can also interpret 3D structures of molecules. And last but very much not least, when you can't or don't want to isolate molecules or don't want to make crystals, then basically what you have is a pleomorphic object. And in that case, you will not be able to take hundreds of thousands of pictures of different cells and reconstruct them in 3D. You have to do tomography, where we take many pictures of the same cell. So here's an example of what we do. It's now old, but kind of like oldie but goodie movie that shows you the process. These are viruses, but you can imagine the same thing going for cells. So you have viruses. We flash freeze them in liquid ethane to liquid nitrogen temperature. This happens about 30,000 kelvins per second. The water freezes so fast, it doesn't have time to form crystals. So it actually looks vitreous or disorganized, just like liquid water. And then we put them in the electron microscope, and now we take pictures of the same sample in different orientations. So I told you each one of these images is a projection through the entire structure. So when you have them at different orientations in this way, this is exactly the same as a CAT scan, except we use electrons instead of X-rays. And our small samples, 1,000,000 times smaller than you, are better behaved because they're frozen. And also now we have all of these projections. For those of the math inclined, if one of these projections corresponds to a plane in Fourier space, so we can go to Fourier space, fill out Fourier space in 3D, and then do a reverse Fourier transform to real space. And what we have is a tomogram, which you will see right now. Sort of it's a sort of a mesh of values where, again, high intensity pixels correspond to low mass and the other way around. And to these data, we can do these kinds of annotations and segmentations to sort of like try to figure out what's what. And again, this, okay, so that's basically tomography. Right. The really nice thing about doing this type of analysis is that if we have enough particles of the same kind, say you're interested in a particular kind of molecule, you can go and find them inside your tomograms and also align them and average them in the same way we do in 2D for single particle cryo-EM, but this time it's in 3D. And you can run classifications and basically get the structure of these things up to really high resolution, depending on the sample and on the abundance of your protein. And then you can also classify. So you can go and say, oh, what is this? You know, if there's conformational changes that are due to function, where are they happening, and how is this part of the cell different than that other one, and things like that. So sort of like a very powerful technique. Now, this all sounds great. And when the resolution revolution came, we were certainly ready. But the issue is that I told you we use transmission electron microscopes, and they have a mean-free path in sort of like this mushy frozen material of about 280 nanometers. Mean-free path means the distance between an electron will go and have at most one interaction with a sample. If it interacts multiple times with the sample, then it doesn't really have information that you can use. So basically things that were more than half a micron were pitch black. This is a mammalian cell. We're growing this in an EM grid. So for those of you that have seen EM before, these are 1 micron holes. They would have hundreds of molecules for single particle cryo-EM. This is an NIH3T3 cell. And you can see sort of like it's a fibroblast, and these little dark regions in the periphery are vesicles. And if we zoomed in, you would see actin filaments protruding and, you know, bringing the cell to places. But as you move thicker or sort of like more and more inside the cell basically becomes too thick and it's pitch black. So for a long time with cryo-ET, we couldn't really do a lot of interesting things. We were looking at isolated mitochondria or just the edges of cells that are very interesting in itself, or even sort of very artificially skinny bacterial cells. So it was really not the thing. And the thing I did in my postdoc, and one of the things that sort of like we use a lot in my lab, is to develop this cryo-electron tomography workflow. When we start by freezing the cells, then we can use fluorescence microscopy to find regions of cells that are of interest for us or that have the particular phenotype. And then we borrow material from material science that I'll tell you in a minute that's called fib milling in order to literally carve little windows in the cell. And then we're able to do tomography of cells. So really what we're always doing is generating and testing structural hypotheses in general context. And then we can sort of integrate a lot of other data available. We're sort of like our image is always as a scaffold or boundary conditions for a simulation, like or similar things. All right. So I'd say I'm from Mexico City originally, and my lab has grown similarly to Mexico City in very random directions. So we have a large part of the lab that works on bacterial cell biology. I'll talk to you about that right now. But we also work with the nuclear periphery. We look at link complexes, nuclear pore complexes, and chromatin. We have a big part of the lab working on the sort of structural architecture of LARC2 in cells. This is a protein that's involved in genetic Parkinson's disease. And I have to say recently, we also added an HIV branch here. But everything that brings us together is just sort of like the methodologies that are what I showed you and others in biophysics that really allow us to sort of advance the field in novel ways. All right, so I'm going to tell you about jumbophage and bacterial cell biology because it's fascinating. And I'm going to try to go fast because we started a little bit late. But this is a very quintessential San Diego thing. There are some phage that are called jumbophage because they're large. And phage are awesome. Phage have given us a lot of tools in molecular biology. They're weird, they're amazing, and normally they're pretty small. And so phages are typically, I don't know, 80 nanometers or so, and they have genomes that are up to, if you think of T4, which is considered a very large phage, that's 180 kilodaltons. But typically lambda, that's the phage that we all love, and all the phages around there are really kind of like less than 50 kilobase pairs. And there's another class that we now call jumbophage that go from 200 to 750 so far. And they're everywhere. And the reason we didn't know they were everywhere is that we literally, when we were phage hunting, people were using 100 nanometer fibers and we just hadn't seen them. So in the era of genomics, we started seeing like all these genomes that are certainly not bacterial, but are too big to be phage. And it turns out there were more phage. So here in San Diego, we found them in aerosol sea spray, in the gut of newborn babies, in a lot of dog poop, in cheese, and in all sorts of places. So if there's bacteria, there's probably going to be a jumbophage around. And I started working on jumbophage because I was down the hall from my dear friend Joe Pogliano, who's, you know, when this phage came out, he started looking at them and he found that one of these phage encoded for a tubulin, like a protein that is, you know, forms microtubules. It's supposed to be in eukaryotic cells. Bacteria don't have it. And here it is, a perfect tubulin analog. And when they're fluorescently tagged, they show that this thing forms filaments. What it does actually, when these phage infect, typically they degrade the bacterial genome. So now it's only phage genome. And then they start replicating the phage. And these things start forming basically microtubules that I'm not going to talk to you about today, but it's also fascinating. And that microtubule center like this ball of replicating phage genomes that totally looks like a mitotic spindle, which is weird. And then eventually they make a lot of phage and they explode. So this is sort of like what was known when I came to UC San Diego, and Joe was showing me this. And when I first came, they were trying to do mass spec to figure out what new, what other proteins are being made a lot during infection that don't go into making phage. Of course, you're going to have a lot of capsids and a lot of tails and things like that. But what else is in there? And the protein that was sort of like really, really abundant was this GP105. And so they decided to basically put a fluorescent tag in GP105, sort of like what you do in cell biology, and look at what that protein was doing. So now here is a picture on the left in red, or sorry, like the dotted line, you can imagine a bacterium. And this is what the fuzzy, this filament that's a microtubule analog or tubulin analog is doing. And the blue is sort of like the dappy kind of like ball of replicating DNA. And this is done in Pseudomonas with a phage called PKC. And this is what the new one looked like, the new protein. So again, the membrane here is in red. Now you're seeing green. This new protein is called GP105, and then the replicating DNA in DAPI. So that is weird, right? Because it looks a lot like a nucleus. But I'm a physicist by training, so I told Joe, well, this is probably the fusion reaction process. You know, this protein is just going, binding DNA and staying there. Nothing to be said, nothing to be done, probably not interesting. But that said, bacterial cell biology is not super satisfying because bacteria are tiny. And therefore, with light microscopy, you can just see, you know, this is like super advanced light microscopy. And so there's not a lot of the mechanism of whatever this protein was doing. And so we decided to take a look. We've never worked on bacteria before, but I had the most amazing student, Kanika Khanna, who figured out how to basically make, oh, I didn't tell you about fib milling. That's bad. Okay, let me go back for a second, because it's a cool movie. Okay, here we go. All right, cryofib milling, really, really quick. What we do is basically, this is a picture of I told you we are a method for material science. These are cells, these are HeLa cells that are grown on EM grids. You see like these little holes. And now we put them in a different microscope. You actually have one in nanothree engineering and we have one in biology. That's called a dual beam. It's called a dual beam because it has a scanning electron microscope and a focus ion beam. And the idea is that the focus ion beam, the scanning electron microscope is, as opposed to a transmission electron microscope that has high energy electrons that go through the sample, the scanning EM basically just sort of like bounces off the sample. You detect the backscattered electrons. And these are the kinds of images that you get, same as, you know, fly eyes or a bed bug in your doctor's office. The focus ion beam, the other beam in this is the same, except it has positively charged ions, so much more massive than electrons. And you can get these kind of images. So this image is actually of HeLa cells in the grid is actually from the focus ion beam. These cells are frozen on the grid. But now you can imagine that if we take this beam that's about 5 to 9 nanometers of focus ions, and we keep targeting it to the same region over and over, you actually start micromachining the cell with exquisite precision. You start ablating layers of atoms at a time. So every time you hear a material science is doing nanopores, you can edit chips and do all sorts of cool things with this. So the trick was adapted to cryo conditions and see if we could use it in mushy frozen materials that are cells. So I'm going to go to this a little bit quick, but this is the microscope, what it looks like. It's a tabletop microscope. There's are the two beams. This is a stage that we designed. Now it's commercially available. And you put there like the little grids, and basically you have the electron and ion beams that are coincidentally. And then you go in and we have this little cell. And we basically use the ion beam to ablate away parts of the cell. And then this is what we call a lamella, and they remain sustained by the sides of the cell. We take the grids out of this one and put it in the transmission electron microscope, like I showed you in the previous movie. So I'm going to skip this slide just for interest. Well, it's kind of cool, so I'll show you. This is what the data looks like. This is a tomogram. I don't know if you can see it very well over there, but it's supposed to be going up and down, and you really see like the dark values are sort of like thicker areas, and we can go and do these annotations of actin on the left, mitochondria in green, chromatin on the right. And again, these cells don't have to be fixed. They're not permeabilized, they're not stained. They don't even have to be GFP tagged. You can take a cell from your skin, make it into an iPS cell, then differentiate it into a dopaminergic neuron, give it Parkinson, and then we can take a look at it if that's what we wanted to do. All right, so like give you a little bit of the idea of the resolution. This is a slice of that tomogram. And here on the left, I hope you can see, we can basically see both leaflets of every membrane. We can see proteins inside microtubules and all sorts of good stuff. Okay, so now I'm going to go back to where I was. All right, so Kanika, who's amazing, figure out how to make a monolayer of bacteria in order to be able to make this lamella, which for some reason that I don't have time to talk about is very tricky. So she managed to infect these bacteria, put them on a grid, and then take a look at them. And the advantage here is that in a bacterium, you have about, you know, you actually catch about the third of the cell and 20 different cells. It's sort of like easy to do better statistics about this stuff. This is the first thing that Kanika saw. So we really wanted to know, what is this protein doing in the periphery of the cell? And as I told you, I just thought it's probably just diffused there, mildly attached. But what she actually saw is this, and I'm going to color it for your entertainment. So in blue is basically what we think is a single layer of protein, no membrane, which is sort of like that protein. We found out later, and the capsids of the phage come and bind to it when they're empty, and then they basically get their DNA injected into them. And these things that you see on the outside in yellow, like little popcorns, those are ribosomes. And so we have the ribosomes on the outside, the genome on the inside replicating. And so that sort of like told us, oh my God, like this thing looks like a nucleus and now maybe it behaves like a nucleus. This means mRNA should be exported and then some proteins, but not others, should come in. So we decided to test this. So the way that we tested this is with Joe's lab. In uninfected cells, we can express a particular protein like REC-A, which is a protein that will bind to DNA. This is a phage protein. So you express it in the uninfected bacteria and then you infect and boom, it gets imported into the nucleus. No protein remains outside. Now, if you see here, GP107, which is a subunit of the RNA polymerase, or one of the RNA polymerases of the phage, same thing, it gets imported. Even proteins from the host, like topo, that are supposed to be inside the quote unquote nucleus, then they go, or then interact with DNA, they get imported into the cell. And conversely, we already knew that ribosomal protein stayed outside, but it was satisfying to see, completely excluded for the nucleus, and then any metabolic protein that we thought to look at, both from the phage or the host, also remains outside. So now this thing really behaves like a nucleus, which is cuckoo for Cocoa Puffs. And if you didn't think that was crazy enough, the entire structure rotates around, which I won't have time to tell you about today. But I'm sort of like going to go a little bit faster. I'm not going to talk to you about this. Sorry, I totally hit the wrong slides. Okay, so what I didn't tell you is that this nucleus is an anti-immune defense compartment. So the thing is, it's one way for the phage to defend itself from the very sophisticated and immune systems that bacteria have, that if you guys are paying attention, are coming out like every week now. So who said bacteria was boring? And so in this particular case, the phage is basically just building a compartment that can import the right things, but prevents things like nucleases from coming in. And so we wanted to know, how does this compartment can assemble without losing its seal? So this is basically like early infection that I'm going to mention to you a little bit at the end. So it goes from something that's 200 nanometers with one genome to something that grows to sometimes 2 microns in size. And all this time, it's not letting nucleases come in. And so this is a picture now of the mature sort of phage. You see a lot of mature phages in here. And it's literally made of a single layer of a 70 kilodalton protein, right? And so that is growing and growing and growing as more phages start replicating. So Tom in my lab decided to take this with subtomogram analysis. He basically used a sort of very sophisticated thing to try to average to gain more information about it. And what he found is that this thing forms actually a square lattice. So that means that it sort of like looks like a pinwheel. So it has a center full fold and a corner full fold, again, made out of this one particular protein. I have to say that at this point, because we were looking at many different phages together with Joe's lab, it was always GP, which is gene product blah, in one phage was a different number on the other, and we were going crazy. So at this point, we decided to name this protein, and we called it chimalin. We think there's enough Greek and Latin names in the literature, so we call it after an Aztec warrior shield that, you know, defends yourself in battle, which is a chimali. So we call it protein chimalin. Okay, so chimalin is weird because nature doesn't normally form tetramers. At every scale of nature, you typically see hexamers and sometimes an occasional pentamer to close the surface. The only place where we could find tetramers was an S layer from Bacillus, but this is embedded in membranes, so you can patch the hole. So we wanted to know how this thing assembles. We contacted my very dear friend, Kevin Corbet, to see if he wanted to do X-ray crystallography. And his very talented postdoc, Amar, tried to do that. And they have very nice SecMals data, but it didn't crystallize. And so Tom decided to look at the void peak. So it's like this B peak over there. And he did see like little shells that were forming. I hope that you can see that through Zoom. And so, but then there was another little shell, another little peak in there. And that peak was 24 subunits. And I told you it's a tetramer. So 24 subunits is 6 tetramers. And that's to our real delight, the smallest surface, closed surface that you can do with a square is a cube. And so Tom had very cute little cubes on the grid that Amar had made. And so then he decided to use single particle cryo-EM to get high resolution structures of this, because we also occasionally do that. And this is the movie. So you can see it's sort of like a tetramer, and it has 4 sides and 2 tops, so to say. And then Tom did something that's called symmetry expansion to basically do focus classification on a tetramer, which increased the resolution considerably. And then he also went ahead and did this on a monomer, and he did this for 2 different species. And what he saw is that there's a core protomer that, crazy enough, had homology to a tRNA acetylase, which has nothing to do with the function of this, but without a catalytic site. And then 2 invading protomers that we call the N-terminus and C-terminus. And you can see here the N-terminus basically binds to the adjacent subunit within the same tetramer, and they're on blue. And then the red one, in this case of the cube, basically does a 3-fold symmetry here to hug, if you want, 2 more tetramers to form a cube. But a rigid body motion of this thing actually then shows you that you can flatten the surface. And this explains how this thing, basically like sort of like a little puzzle piece, can, account for the high curvature and sometimes even negative curvature of this surface while maintaining this thing together. So now we knew basically how this thing was formed at the atomic level, but what we wanted to know was whether it was possible to just transport proteins and mRNA through this thing. And for this, we went to my very dear friend, also Romy Amaro, who's in my department. And basically, Christian Seitz, who's a grad student in her lab, took this entire sheet of 9 tetramers and then tried to do molecular dynamic simulations to see what could be transported. And Christian found that so that the center fold is about 1 to 2 nanometers. You can transport water metabolite, probably could thread mRNA, but not fold the protein. So we're still chasing that down. And I'm just going to finish with telling you, like, we generate as many or more questions than answers. We're studying a lot of different questions about how are proteins imported. We actually have found what we think is a nuclear port that has disordered domains. How does mRNA get exported? How does compartments form? And just to tease your brain, but this drives me a little crazy, I'll show you a very fluke. I mean, it's like kind of like a fluke, but reproducible experiment where in one particular phage and host, eGFP gets imported, which technically shouldn't, but it does. But GFP plus and superfolder GFP do not. So we're talking here like 1 or 2 point mutations in the loop of a beta barrel that will allow a protein to get in or not. And we can tag things to eGFP and they will also get imported. So exquisite precision with this compartment. Okay, I don't want to tell you much more about this. It gets a little bit crazier, but I want to give chance to the other student to talk. Just want to say, we put together with Joe and Kevin and Kit, like a lot of different techniques that we basically apply to this process. It's a lot of fun because we're typically, I didn't even show you like some FRAP data and stuff, and we're just like looking at things like, for example, this phage that seems to have a genome organized inside the nucleus that then gets, like you see here, a single molecule of DNA going into the phage. So what is this protein? And then we go and chase it down, and then it ends up being like a weird thing. One last thing I'm going to tell you is that we have a phage class at UCSD. Gina was an amazing undergrad in Joe's lab, and Amy is a joint student between us who was doing TAing at Joe's class. And in this class, they basically, it was during the pandemic, so what they did was just phylogenetic comparison of the phage and found that only about 30 or 70, depends on what you're looking at, of 300 to 400 proteins are conserved. This actually led us to think that this is a really large phage. Again, T4 is a very large phage, and you can see like the difference. This is a ribosome for scale. And it doesn't seem like all their genomes are conserved. So now normally phage are lean, mean killing machines, and you can't engineer them because if you take something out, you break something else. But this phage actually looks like it has room for error and 2 other things. So we decided that we were going to try to figure out what the minimal phage is, engineer payloads into it, so like anti-immune factors, maybe even antibiotic synthesis, broaden the host recognition, among others, train this phage for particularly nasty infections, and then basically build a toolbox for individualized bacteriophage therapeutics. So we put kind of like a dream team together. These are the people that I talked to you about on the left. We have the Craig Venter Institute, Justin Meyer, who's an amazing evolutionary biologist that does this sort of like phage bacteria evolutionary analysis. Paul Turner and David Pride are also amazing scientists who also represent 2 phage centers in the country, including IPAC here at UCSD. And we have funding for this to really bring it all the way to preclinical with for antibiotic resistant infections. So we're trying to get to preclinical for cystic fibrosis in 3 years. So with that, I have to say this is my lab. I'm super lucky in blue, sort of like all the people that worked on phage. I have the best lab with creative, fun, kind, awesome scientists that I have the pleasure to work with. And of course, I've done all the work that I showed you today. And with that, I just like to thank you for your attention. Always looking for exciting bioengineering students. We do a lot of methods development. We need microfluidic devices to optimize this infection, freeze in different ways, a lot of engineering, a lot of cool delivery systems to cells and other stuff. And of course, really cool biology. All right, thank you. And sorry I took so long.
Thank you, Dr. B. I'm so sorry for rushing you, but thank you so much.
No, it's okay.
We're gonna have you back and
Yes.
do an entire session and have time to ask questions. So thank you so much. And I will tell you all that Christian sites, is a graduate of the Interfaces program.
Yes.
Yes.
I
So
yes,
yes, he got the PhD specialization and he's doing some fun stuff with it, looks like. So thank you again so much. Sure do appreciate it. And I don't even know how to transition you to Just Don's presentation.
I think I'll just let you, and then if you guys have any questions, email me, or if you want to come hang out, see the lab, the microscopes or whatever, I'd be very, very happy. And then, yeah, make sure to tell new students that we're very big fans of the Interfaces program. And
Yay, thank you.
all right, cool.
Thank you. Tell Rimbaugh hi for us. She's dog sitting today. For those of you who don't know, Just Don is a 3rd year, right?
3rd year, yeah.
3rd year, PhD, biomedical sciences. Yes, I get biomedical and biological mixed up. And he's working on trying to dissuade us from eating red meat. So hence our snacks today. For those of you who have not availed yourself, please do. Is it recording? Can you tell?
Only 3 dogs. If you're supposed to screenshot it on the far right. Oh, host disabled participants can share.
Oh, that's right. Yeah, because, and I couldn't share it with you. Thank you. Yeah, because your address is health and it wouldn't let me add you as a host. Dr. Villa, can you hear us? We can't hear you.
Guys, that's because I was muted. Yes, so
Can you
I did.
continue?
Yes, I
Can
will.
you
I will.
continue to host.
I'm leaving, but I will. So now you should be able to share, and then I'm going to make you host.
We'll work it from this end. Thank you.
Yeah. Okay. Sorry about that. Thanks, guys.
Thank you. Except we can't do that because
I think we're good.
I think we just keep going.
Yeah, we're all good.
Oh, yay. Oh, goodness.
Thank you for that introduction. I'm gonna go through this really quickly, so probably gonna skip over a lot, but that's okay, because if you have questions, I'm happy to talk about it after. And this is really an extension of the conversation I had last year. And so when Kelly reached out to me to speak, I had just finished up a massive review that had been started in my lab about 10 years ago. And I was like, okay, this seems additive to the story that I'm going to tell. So I'm going to talk about red meat briefly and the role of human evolution, and then explain why it might have some issues that might be important for those who study biological sciences or health and disease. And so, just so we're all on the same page, lots of definitions of meat in which they use to study it in human evolution. And with respect to epidemiology, it's very different nutritionally from what they define it as culinary. So in the culinary arts, red meat is considered red when raw and maintains a dark color after it's cooked. And white meat is considered pale in color when raw or after it's cooked. And so due to some successful marketing campaigns, people still believe today that pork is a white meat that started, I think, in the 1980s, late 80s. But no, red meat is, it's defined as any meat of mammalian origin, which is based on the myoglobin content of the meat itself. So we're all on the same page. Okay. So as you all know, we live in a world that is increasingly consuming more and more red meat. I really like this picture here at a grocery market, which demonstrates the abundance of poultry options, which are here on this far side. And the rest of the supermarket is dedicated towards red meat. So in the US, the most consumed meat product is red meat. And it is decreasing in a couple of countries, but in general, the trend for red meat consumption is increasing worldwide. It is illustrated in several TV cooking shows. It's included in many cooking glorified channels on Instagram. I don't know if any of you know the raw red meat eater, but he's got millions of followers. It's a definite delicacy item, and there are several books written about it. Several in the past few years about why you need to eat lots of red meat. And I just want to contextualize it a little bit by saying that while the concentration of consumption is increasing, I think what we're starting to see is a real bottleneck here, and it's starting to confine its own growth. So generally in a worldwide trend, you see the increase over time, but at this, as red meat consumption increases, the prices also increase as well. So what that means is it's getting more expensive to produce this amount of meat for consumption with highly infectious agents when animals are confined in tight spaces, when there are also extreme weather events too that impact the growth of the feed crops. And the feed crops have also plateaued. So we might be seeing the first signs here that this system is no longer going to be super sustainable because prices reached an all-time high in June of 2022. With respect to the greenhouse gas effect and the emissions that are released from farming in these sites, it's not negligible. What we're witnessing here is lots of different effects with the greenhouse gases. And I basically entered a wormhole this past winter looking into all of the different ways in which people model greenhouse effect warming. And it's really complicated. It's really complicated science. But essentially, carbon dioxide is a long-lasting greenhouse gas, which can last in the atmosphere for hundreds of years, if not centuries, whereas methane lasts about a decade to 2 decades at most, but it's extremely potent. So the heating effect is much greater for methane, which is important because a lot of the red meat animals, ruminants, mammals, are the big producers, and that's not through flatulence, it's through the digestion process. So they don't have 4 stomachs. For cattle, for example, they have like 4 different compartments. And so that process of digestion allows them to belch, and that is what causes the methane increase. So that's just some, oh, and this is another, so this is, I wanted to show this one first because it's just by virtue of being humans and using the land, it's going to cause some warming effect with the massive scale in which we're producing food. Chocolate, for example, I was pretty surprised to see that high up. Rice is up there too. That's because it's grown in like, I think, fields that are wet and release a lot of methane. So there are some vegetable options and non-meat options that are contributing to the effect as well. But if you stratify it and look just at protein sources, you begin to see that it's a little bit clearer here that the impact for the climate is always going to be highest for mammalian sources. So for example, if excluding all methane completely from lamb and beef, you're still going to see a higher greenhouse effect, 5 times higher compared to tofu, 10 times higher for beans, 20 times higher for peas. So the conclusion is that meat and dairy still top the list and differences remain large. And I really wanted to emphasize that because that conversation is also not really factored in to the international discourse. If you look at the Paris Agreement, only a third of countries that are registered in the Paris Agreement even factor agricultural as a factor worth addressing in the 21st century. So we're in a conundrum. We have 100 billion people, or 10 billion people we're going to have to feed by 2050. And we're seeing massive increase intake in meats in middle and low income countries. And that is, I think, best illustrated by the fact of what's going on in the Amazon rainforest, where this century alone, 60% of the forest in the state has been cleared for cattle pastures. So those are the Earth's lungs, and I think that's a really important factor that is not being considered here in the larger context of what this means for health trends, health projections. All of this modeling sometimes do not factor in these equations. So with that being said, I wanted to briefly talk about red meat and human origins. And I wanted to very clearly say that there's little dispute that animal tissue, so lean muscle, bone marrow, brain tissue, were essential components in the evolving diets of the ancestral hominins around 2.6 to 3 million years ago, if not earlier. But I also wanted to say that any ideal diet is very context dependent. Where did your ancestors move? Were they in Africa? Did they migrate up north to the Arctic and depend on seal fat, for example? And so ideal species adapts to a diet that is one that promotes health and reproductive success. And humans have been very fortunate in that we're very adaptable, and the dietary variation is inherent to our kind and perhaps to the entire lineage. So each one of these brackets could be its own discussion and content, but the reason that there is so much emphasis on red meat consumption throughout human evolution has been the fact that it's one of the most nutritionally dense foods available. We're talking all of the essential amino acids, the conditionally essential amino acids. We're talking about protein content. We're talking about cholesterol, which is needed for every cell in the body. Available heme and heme iron, which is a really interesting topic I would love to talk about more, but not today. And then other macronutrients as well. So it's a tightly knit package of several things that the body needs to function. So the question really is has red meat been overemphasized? So there's two camps here. One of the camps is that red meat consumption has served as a catalyst for several key transitions in human evolution. So it was involved in pair bonding, tool making, brain expansion, cooperation. These are the leading theories in the field, and they have been persistent throughout time and increased longevity. So the more nutrients that you get, better survival, better health. Okay, but there's an entirely new camp that's saying, hey, wait a minute. It's also possible that there's superior preservation of animal bones and stones compared to the fibrous plant material. So we have a preservation bias here, right? The history is written on the stuff that's left behind. And so the things that we know to generate health, which include fiber, don't really last. Even resistant starch plant material does not fossilize well in comparison to animal ruminants. So the stones, the tools, the bones, fossils. And through this investigation, the oldest food container that we know of is around 13,000 years old, in which they use to ferment food. So this is long after humans have evolved. So we'll probably never have a clear answer to like what came first. And that's a whole other talk in and of itself about, is it truly the meat consumption that made us human, or was it the fermentation of food and that unlocking the same vitamins, minerals, and nutrients in red meat? Or was it that we cooked it, but also at the same time, isn't fermentation just another modality to cook? So really interesting conversation that I can't get into today. But I also really wanted to emphasize here that what is also underemphasized is the pivotal role that women and children have played as consistent providers in daily fiber-based sustenance. Because when earlier hominins were gathering food, the fiber-rich components were a much more stable source of food than meat, which was kind of come and go whether or not the tribes were successful in hunting down whatever it is that they were trying to catch. So does red meat still provide a fitness advantage? If you look at any meta-analysis, the answer is starting to say probably not, because of the associations we see with high red meat consumption and colorectal cancer, type 2 diabetes, stroke, so cardiovascular disease, and essentially excess BMI and all-cause mortality, which is the key one here. And so not only are these the ones that are most pinpointed, they are also the leading causes of death worldwide. And these conditions were nearly non-existent among hunter and gatherer societies who we assume to have consumed lots of red meat throughout time, depending on locations and where you were. But specifically, when I joined my lab, I was really interested in colorectal cancer. And so if you look at the Eat Lancet report from Walter Willett at Harvard, he showed that, and his colleagues showed that if you want to talk about the highest increased risk with the most evidence and the most convincing association, you're going to see that colorectal cancer is all, besides one, they're all dietary related. So meat consumption, excess alcohol consumption, processed meats, elevated BMI, and adult attained height, which is just another way to say the higher you are, the taller you are, the more likely you are to develop colorectal cancer. Which I think is probably related to the fact that you just have to energetically introduce more nutrients and calories into cells that are probably starved for nutrients. And if you're feeding them without the prepackaged other things that came along in the human diet, you're missing a lot. So what's here that I wanted to mention is that it's so hard to tease apart the association between red meat consumption and chronic disease, because when you increase red meat, you're also decreasing fiber, and increasing red meat also comes with associations that are associated with increase in risk of the same thing. So you're more likely to smoke, you're more likely to have a sedentary lifestyle. So it's unfair to cause one, but when one shift happens, there are multiple reasons that the outcome, the colorectal cancer outcomes we see are a reflection of multiple changes. So these are the confounders, but they all kind of flow in the same vein. So the associations here are strongest for colorectal cancer, as I previously said. And adherence to a Western diet post-diagnosis shows that it's associated with a higher recurrence mortality. So if you have colorectal cancer, you get treated, you're cured, and you go back to your same lifestyle, you might see it again. So what's going on here? What is this? What's the reason for this? And all of these conditions have one thing in common, which is chronic inflammation, which is a driver of a lot of chronic disease in the US and in the world. So we know there's a lot of inflammatory markers circulating antibodies against something, but what is that, for example? So I'm going to be very quick here and show that there are lots of different theories about what it is in red meat that is so inflammatory for humans. And if you look into a lot of the theories, none of them are specific to humans or they're specific to red meat. But one that for sure is sialic acids, glycans, or glycobiology, which is what my lab studies. And so very briefly, this is kind of the focus of my project and what my lab's life's work is on. And it's on the glycocalyx and all of the different decorations that happen on the outside of the cell. And this structure is so important that in 3.5 billion years of evolution, life has never generated a cell devoid of extensive complex glycosylation. Molecularly, that looks like this. And to be even more specific, what I focus on are the little diamonds. They are the sialic acids. And here is so small. But basically on the outside of these cells, we have a way of the body to self-recognize itself. And so what humans have are a little molecule called Nu5AC. And then in lots of other mammals, they have another sialic acid called Nu5GC. And so Nu5GC is completely absent in humans. We don't express it naturally because throughout human evolution, around 2 million years ago, we lost the enzyme that produces Nu5GC, so that other diamond on the outside of the cell surface. Lots of different theories for that, can't get into it today, but essentially when that's wiped out, this entire cell surface is completely changed. So it goes from being this kind of hydrophilic cell surface to a very slick and oily surface. And so that's what our cells look like on the outside. We don't have this glycolated group on the sialic acid. And so again, here is the enzyme code, and the dashes here is just the chunk that fell out. And that that loss has resulted in an enzyme that's no longer functionally active. And so we still make the enzyme, it just doesn't convert that sialic acid. So our cell surfaces look very different. And again, this is only a single oxygen change here. So this is highlighted in blue to show the difference here, that what is absent from our cell surface naturally. Humans are not the only one that's lost the sialic acid. It's been lost in reptiles for over 100 million years. The New World monkeys have lost it as well. And so, interestingly, bats too have lost it. So when you talk about bats that are cultivating viruses, for example, and they're shedding them out without any new 5GC, it'll look more like self, which I think is really fascinating. But again, can't get into that today. And what I like to kind of equate it to is that think of the food that you eat as having an additional thing on a nutrition fact sheet where it lists a sialic acid content that may be sensitive or reactive for you. Importantly, sialic acids don't really add much caloric intake, I don't think at all. But that would be a way to quantify, like, you're eating a food with a high amount of a foreign sugar that is very inflammatory. So here we see that the highest amounts of this foreign sugar that's inflammatory is from beef or steak. Also really high in fish, like specifically the fish eggs, so caviar, really, really high. So, but once they reach adulthood, the enzyme turns off. So there's a temporal regulation to this enzyme too, which is really fascinating. And it's tissue specific too. So that's again, whole other conversation for a whole other day. But in summary, it can be boiled down to this. When we consume animal products, probably not so much milk and butter, very, very low in GC. But when you get this molecule from the food, we have an evolutionary holdover of the enzymes that can put it on the cell surfaces. So again, we don't express this naturally, but when we present it on the cell surface, it is reactive. The immune system sees it as that's in a different sialic acid that has an oxygen on it. It's different. It is not self. And so once you have incorporation, you have antibodies that are directed towards it, and hence the term xeno-autoantigen, xeno-foreign, auto, self-antigen, self-antigen, okay? So the antibodies to that are xeno-autoantibodies, which results in a condition, low-grade chronic inflammation known as xenosylitis. Importantly, this is not found in reptiles, so chicken, poultry, and it's completely, all sialic acids are absent in plants and fruits and veggies. We know that when we feed this, for example, when we feed it to mice, the human-like mice that we've knocked out the gene, we see that it is incorporated preferentially into epithelial and endothelial tissues. And so if we inject tumors, for example, we also see increased growth. So remember that study that I showed, or the association I showed earlier, where if you have colorectal cancer, you get it removed, treated, you go back to a Western diet, you're more likely to have recurrence. So this is just showing that there is some kind of hypoxic condition in cancer, which kind of upregulates some of the enzymes to kick that foreign sugar when it's consumed in the diet to the cell surface so that it's presented for the immune system to eventually recognize it. Recently, so right before I joined the lab, there was a paper that came out in 2021 that showed even in the context of a high-fat diet, which, we have, historically thought that all like high-fat diets are really bad, really inflammatory, but we've actually learned now that if you feed high-fat diets and time restricted, it's actually anti-inflammatory. So there's a time component here, but that's a completely separate mechanism. But with respect to this foreign sugar, when you're consuming it in a high-fat diet, the results are even worse. And so what does the Western diet usually contain? High fat, high sugar, and more meat, less fiber. So basically, when I was here last year and I talked about my research, I was really interested in looking at the first ever biomarker for red meat consumption to determine how much red meat you're actually eating. So it'd be taking a serum sample my project has shifted a little bit because we might not have the funds to do all of that mass spec analysis, which would cost thousands and thousands of dollars. So I'm focusing right now on a mouse model to understand at what point does this foreign sugar in the diet become inflammatory? When does it start to tip over and become pro-inflammatory? So the question really is, what is the threshold percent of GC that induces this phenomenon known as xenocellulitis? Because we know that every human tested so far has antibodies directed against this foreign sugar, and this exposure probably happens pretty early in life through bacteria in the gut, so the microbiome, maybe it's taking up this new 5-GC, foreign sialic acid, expressing it. Immune system says, hey, that's not you. We eat it later in life. And depending on how you initially mounted an immune response may determine how pro-inflammatory red meat is for you and what that might mean for tailoring a diet for you in the future to decrease inflammation. So if you eat meat 3 times a day, you might want to consider reducing it if you have more antibodies against it, because the outcomes seem to be a little bit more worse. So I'm doing a mouse model right now, trying to determine that. All I wanted to show here is that I'm feeding mice high amounts of this foreign sugar, a 1-to-1 ratio. So the AC is pretty protective. So the AC would be poultry, for example, or cheese for another mammalian example. Previously, what has been used is edible bird's nest. So edible bird's nest is a luxury dish consumed in China. It's very, very expensive, but it's loaded with new 5-AC, and it's been I don't think there's any large study to show it, but it's been a longly used traditional food in traditional Chinese medicine to help prevent the flu, and they take it when people are sick, because the thought is now that the AC might kind of prevent some of that inflammation. So basically, over the course of their life, I didn't have time to create a better graphic for this, but over the course of their life, the mice in the high fat, the high new 5-GC diet are going to be exposed to the most GC, and the diet at the lowest amount of GC, they're only going to experience about 0.5. But I'm really interested to see what's going to happen to this 1-to-1 ratio. I think in my head when I originally started or conceived this project, I think in my head I thought that as you increase, there's going to be a gradient of more inflammation, more inflammatory markers. But there may be a tipping point where it's protective, and once you cross, maybe it's the 1-to-1, or maybe it's 75-to-25, maybe that's when it plateaus and you see this cross. So I don't know what's going to happen, but I've set up the mouse model to start immunizing them against this compound, so then they can eat the food. Again, making the prep for the diets took 2.5 months, and then quantifying how much AC was in each sample took a long time. So this is very much new, and I'm at time here, but there's lots of different ways to take this project, and I'm really hopeful that another avenue comes, knowing that we, in addition to presenting this on cell surfaces, it's also metabolized, and so when it's metabolized, we can actually detect that too. And very, very interestingly, this metabolite can be preserved in fossils. So the reason I brought this up and talked about human evolutions in the beginning was because when I think about program interfaces, it's multiple scales of biology, and I would argue through multiple planes of time. And so using our knowledge that this foreign sugar can be metabolized and be preserved in fossils, we can actually determine what other early hominins are more similar to us, compositionally speaking, versus ones that are further away. So what that means is, in answering the question, which early hominins led up to Homo sapiens, we can actually have a better estimate of that using the knowledge of biochemistry, metabolism, this pathway, our evolutionary origins, and importantly, the fact that when you knock this out in mice, all of these metabolites, when you knock out the gene that produces the sialic acid, when you knock that out, they don't have that in their sample. They don't have that in the serum anymore. Similar to humans, except for humans who likely consume red meat. They're going to have trace amounts. So if we see something with no or trace, it's a signal that, okay, red meat was possibly consumed in this person's diet. But if you don't detect anything, then it's very likely that what you're looking at is an organism that had a functional intact gene, which again, we lost about 2 million years ago. So I'm really hopeful that I can get my hands on some bones, some ancient fossils, or collagen extracts to look at this as well. Because when we do that, and it's a very simple technique. You extract it, you treat it, you filter it, and then you run the samples on mass spec. You treat it with chondroitinates to chop up the disaccharides so you can have a good signal. And then once you have that, you can actually kind of start to make sense of who leads up to who, which I think is really exciting, and I'm hopeful. We'll find out if one of our collaborators who applied to a grant this past year, she finds out the status of that this month, I believe. So if that's the case, I think we'll get around, I think UCSD will get around $200,000 to process these samples, which again, on fancy mass spec is really expensive to run per sample. You're looking at $200 to $300 with knowledge and expertise in the lab. And yeah, so I do think that with respect to scales of multi-scale biology, I'm looking at a single oxygen atom on a single sialic acid measured around 1 nanometer, and they'll be like, okay, look, so this really tiny thing, but which ultimately leads to complex disease process through antibodies, and leads to associations of epidemiology in which we see with colorectal cancer and heart disease, human evolution, the study of fossils, and even climate science and planetary change. We know that what we're doing right now is not sustainable, and even if we stop all greenhouse gas emissions, the planet will continue to warm for another year. So I do feel like my project kind of sits at the axis of so many intersecting things and so many experiences that I had in my life, because my undergrad was in environmental science. And so being in a biomedical science program, it's been really fun to see how I've been able to fuse it all together into kind of a complete, coherent package of information for our kind and for our species for the future. So with that, I went over 15 minutes, but thank you for saying I think you thought it was interesting NC State. So I really appreciate it. Thank you.
I wish we had more time. So sorry. And thank you for your attention. And please make sure you sign in. Oh, Marcus, would